itself like this. This is a lipid molecule and a virus has been wrapped inside. Now, it goes outside the infected cell. The home from which I came to you, that home has been infected by a virus. Please have some preparation. This is the news that has been spread by the interferons. Okay, welcome to Ibrahim Academia. Today we are going to discuss the infection of HCV, that how HCV virus enters our liver cells and how this causes infection inside. What is the entry mechanism of the HCV inside the hepatocyte and what it does inside the hepatocyte, how the machinery of hepatocyte has been used for the assembling and for protein synthesis of HCV. All these things will be discussed in this lecture. The hepatitis C virus is a positive single-stranded RNA virus. What is meant by the positive single-stranded RNA? Means whenever this RNA enters the hepatocytes, the liver cells, this RNA can directly be translated into proteins. There is no need to make its complementary side, to make its negative RNA. No, you can directly use it as just like messenger RNA of our body. Another important thing is that HCV is enveloped virus, the outermost boundary. This is envelope. This is a lipid, and from where this lipid comes? This lipid is actually the lipid of my own cells. This lipid is the part of my endoplasmic reticulum lipid. Now let's talk about the normal mechanism of viral infection. Whenever there is virus, so this virus enters my body cells, any kind of cell, this enters by endocytosis, and when the capsid and envelope dissolves the genetic material, that could be RNA or DNA, there is some kind of protein which is called RIG, retinoic acid inducible gene 1. This protein resides inside my body cells. This protein senses this genetic material of virus, and when it senses this genetic material, it gets activated, and after its activation, it runs towards the mitochondria. On the mitochondrial surface, there is another protein embedded which is called MAVS, mitochondrial antiviral signaling protein. This MAVS gets activated, and after that, there is a cascade, the phosphorylation of 4 or 5 proteins, and after that, a transcription factor gets activated which is called necrosis factor kappa B, NFKB. This NFKB travels towards the nucleus of my body cells. Inside the nucleus, it activates some kind of genes which then translate proteins are called interferons. These proteins which are called interferon alpha and beta, these interferons class 1, they comes out of the infected cells and spread the news in the surrounding. It spread the news to its brothers, the healthier brothers, that, oh brothers, we have been infected. There is an infected cell. Please prepare for a viral attack. The home from which I came to you, that home has been infected by a virus. Please have some preparation. This is the news that has been spread by the interferons. And the interferon, what actually happens molecularly, there is a Jake state pathway activation in the neighboring cells. After that news spread by the interferons, the neighboring cells prepare some antiviral proteins. Among those, there are some proteins that can block the translation of viral proteins, and also a special kind of protein which is called major histocompatibility complex type 1, MHC1. The overexpression of MHC1 occurs upon cells, and this overexpression of MHC1 molecules upon the surface of my own cells indicate that here is some problem. We are not feeling well. My body's immune cells come and they say, oh, how many MHC molecules have been there upon your surface? What happens, my brother? The MHC, the overexpression is a signal that there is a viral infection. This was a normal mechanism of a viral infection, but in case of HCV, this not happens. HCV has some other special kind of mechanism because it has some proteins. It has 10 proteins, 10. And among those 10, there is a protein which is called NS3. This NS3 is protease. Protease means the cutting of protein. It can cut any protein. It has a protease activity. When this protein, this virus enters inside the cell, the NS3 protein of HCV come and it deteriorate or cut this MAVS protein on the mitochondria of my own cell. And when MAVS protein has been cut by NS3 protein of HCV, the entire signal blocks. As the HCV virus attacked the hepatocyte, it entered inside the hepatocyte. Inside hepatocyte, it has NS protein which block the MAVS. The entire signaling has been blocked, and this virus start the replication or the duplication of its genetic code, which is RNA. It also encodes proteins for its assembly on the endoplasmic reticulum. On the smooth endoplasmic reticulum, it gets the assembly of its entire protein, its entire structure, and on the rough endoplasmic reticulum it synthesizes its own proteins. And after that, when the entire assembly has been completed for all the new viral particles, the virus then uses a trick. Now this virus molecule has to move outside of this hepatocyte, this infected cell, and it has to infect more healthier cells. This virus, when it exits, it takes some lipid molecules along itself and it wraps itself inside that lipid molecules. Let me give you an example. This is lipid molecules, a lipid bilayer of my own cell, the endoplasmic reticulum lipids. The virus wraps inside itself like this. This is a lipid molecule and virus has been wrapped inside. Now it goes outside the infected cell, and in the interstitial fluid, my immune cells can't recognize this virus. They think that it is my own lipid molecules, it is my own cells. They can't recognize because of this lipid molecules. That is why enveloped viruses are dangerous. Somehow they are dangerous because they cannot be recognized easily by my own immune cells. The HCV symptoms appear very late. It takes months, years to be detected. Then the hepatitis C virus, when takes full grip upon the hepatocytes, the liver cells, then some special kind of liver cells, they form scares outside of the cells. They contain collagen type 1 and type 2, and because of these scares, the supply to the cells interrupts, and this scares formation is called fibrosis. And after the fibrosis, the condition may worsen after some time, and then this leads towards another chronic condition which is called cirrhosis. Cirrhosis is irreversible. After cirrhosis, the liver may experience carcinoma. My own immune cells are very much intelligent and specific in operation, especially the T cells and the B cells. Whenever they called by macrophages or the dendritic cells, that we detect someone, this is not our body particle. Oh, how is this possible? Wait for us, we are coming. The B cells and the T cells came and they detect this particle. They then start the antibody production. The B cells start some specific kind of antibodies that can only fit to this structure. For example, this is a B cell antibody for this specific structure. Say this is E2 antigen of HCV. This fits. Now, but the problem is that HCV is a RNA virus, and RNA virus starts mutation in hours. It mutates itself. It changes the genetic material, the genetic information. And when the genes changes, so genes will definitely code for the protein, the entire protein changes. For example, after an hour, the E2 antigen adopts this shape. Now, the B cell or the sons of the B cells are now in the preparation of this kind of antibody. Now, tell me that is this structure can fit into this antigen? No, this cannot be fit because this is now a separate structure. This antigen is now changed. This antibody can't work against this. No opsonization, no agglutination process can happen now. Now this antibody is calling its mother, its B cell, oh mom, the shape is changed. This is a wrong attachment for me. This is calling again to its son. Oh baby, no, this is the same structure. I have examined this 20 years before. No, ma'am. The shape has been changed. Oh, how is this possible? Now that B cell starts another type of antibody, but not that specific B cells. Its sons will then start. This antibody will be prepared by the offsprings of the B cells. This kind of antibody. After 2 hours, this shape is now again changing because there is changes inside the genetic material. This shape is now has been changed from rectangular to like say this one. This kind of shape is now. Now, can this shape be fit into this type of antibody? No. This antibody again will not work. This is now not working. This is not working. Now, this antigen needs a specific type of antibody that can fit into it. The B cell will now again proliferate to form specific antibodies. Now, the B cell will divide. They will form new antibodies. But there will be no memory. No memory for the HCV. That's why HCV is dangerous for the health. Thank you very much.